My co-founder, Matt, he had this idea far away in New Zealand, and I was in England at the time. And he found me randomly on LinkedIn and reached out and was looking for someone of my similar technical background. He had this high level idea that, hey, dairy products are nothing else but coagulates of some dairy protein. And like, what if you can make this protein out of animals? We don't need a whole milk system. Then if you make them from, say, genetically engineered microbes and you purify them out, well, you could make phenomenal dairy product because structurally these proteins are key to what gives you, say, a cheese eating experience or yogurt eating experience. We got accepted to IndieBio, and that really propelled us, gave us a lab and money and a place where we could develop a lot of things in just a few months and raise our first round of funding after. The good thing to do is not to innovate, to use what's existing out there. That is the cheapest way. We had to learn this the hard way because, in the beginning, we maybe did do things that were novel and different, but then were not easy to implement in a manufacturing side. We do like to say that our cheese is for everyone. This is a cheese that brings everyone to the same table, whether you are lactose intolerant, health conscious, care about sustainability, vegan, and you want to eat tasty and nutritious cheese. Everyone can eat this cheese, really. And that's the difference to the products that are in the market.
Yeah, thank you so much for coming to the Biofounders podcast. It's great to have you. I've been following New Culture for some time, and it'll be very interesting to have a chat.
Thank you, Sophia. We're very excited to be part of your podcast too.
Great. So I was looking at your LinkedIn, obviously, and one of the things that caught my eye was you used to do other kinds of research completely unrelated to new culture as it is now. And one of those areas was marine biology, if I remember well. So tell me about, your former interest in biotechnology and if you've been like just out of personal curiosity, following any of those spaces recently?
Sure. Yeah, I studied biology. I studied molecular biology as at undergrad, but on the side, as my kind of passion projects and courses, they were all in marine biology. I really love sea. I grew up on the seaside in Croatia, very connected to the sea, and I really started diving and doing research in the sea mostly because I love the animals. But that was always something that was just my hobby and passion, and something I did on the side while studying. And then my main course was molecular biology, and I was always fascinated and obsessed with proteins, as I think we molecular scientists are. And that journey led me to a lot of research on protein structure and function. And a lot of it was in vitro work, meaning you would, work with proteins as purified molecules and study them in various ways, structurally or functionally. And that's when I did my master's. I moved to Switzerland for my master's, did my master's in Zurich at ETH, and it was focused on protein structure and biophysics, and I absolutely loved it. But I realized I missed in vivo work. I missed like biology. And that really told me, hey, I actually am a biologist, which was first time in my life kind of clear to me that, even though I love all life sciences, so that led me to do my PhD in something again quite different. I moved to Cambridge and I joined Jason Chin's lab, who is kind of like a pioneer in many areas of synthetic biology, including orthogonal ribosomes, unnatural amino acids, now genome, genetic code expansion, and genome modification and expansion methods. So yeah, so my PhD was actually quite interesting. I was in vivo in tiny, tiny little worms, these nematodes, C. elegans. I was able to modify a particular class of proteins with these methods that my lab was developing. And the goal was that, we really struggle today, if application was in neuroscience, we really struggle to understand neuroconnectome well, even at very simple neuroconnectome levels, such as in those little worms, where we know each neuron basically. And the idea was to have a method that enables us to, like a switch, turn on and turn off individual neurons, like single neuron at a time. So to trigger activity or inactivity of a single neuron and study how would that affect behavior of the worm, like what will that do when the animal's like going towards, say, food or reversing from something they don't like, and how will like interfering with their nervous system, basically to that molecular precision, what it
Did
would
that
do.
involve any kind of optogenetic systems, or how
Yes,
did you actually?
absolutely. That's it.
Okay,
My PhD was in optogenetics. Wow. Well,
very
yeah, great.
interesting.
So my PhD was a particular application of optogenetics where, we were able to, yeah, trigger these expression of these optogenetic channels only in single neurons by the way that we were controlling a recombinase by light, where we were incorporating these unnatural amino acids that are light controlled. So that would then allow me to actually, have live animals freely moving in like an imaging system that we built. And we would then, when we would shine a light, we would activate optogenetic channel only in single cell, basically, which was like until that time, that was never done. That was never possible, to be done. So yeah, so that was very exciting to me. It was very challenging. Obviously, these systems are very complex. They're very different to like few proteins in a buffer
Mhm.
isolated. Um, that's kind of how my journey went, and I stayed to do a short postdoc in the same area, that kind of synthetic neurobiology in C. elegans, and after that, I actually left academia. I thought academia wasn't for me. I loved science, but I wasn't gonna become a professor or stay an academic. And so, how you asked me, how did I end up making cheese? After I studied, worms and their neurons. And the thing that it has in common is the protein, really. It's the protein
Sure.
science, right? Again, all my love and research is about proteins, understanding and studying proteins. But new culture is also my like kind of private pet project and my personal passion, because I did become it was about 10 years ago, I started becoming really obsessed with climate change, not in a good way, obviously, kind of in a bad way, where I was very aware of it, and I was very aware of sustainability issues we were facing. And as I was reading and researching more, obviously this was not in my own field, where I would be more interested in drugs and therapeutics, and which I all follow and love. But this was more like I wanted to understand how the world works, and why do we have this problem, and how are we contributing to it. And I really became kind of obsessed with what can I change in my private life. I went vegetarian, I tried to become vegan, and I always struggled giving up cheese and eggs. Those are the two things I always struggled with. And I tried to live like zero waste for a year, literally produce, minimum waste. I tried to change how I buy things, that I buy very little, and all these things. But then I realized, hey, I'm just one person, n equals one. Like, what does this matter? I'm looking like some crazy lady. And how can I work in this area? How can I contribute to actually reducing carbon footprint globally and to making world more sustainable? And so New Culture was a very fun and serendipitous story, because my co-founder Matt, he had this idea far away in New Zealand, and I was in England at the time. And he, found me randomly on LinkedIn, and reached out and was looking for someone of my similar technical background. And he had this high level idea that, hey, dairy products are nothing else but like coagulates of some dairy protein. It's protein in milk. And like, what if you can make this protein out of animals? Like, you don't need a whole milk system. Then if you make them out of animals, which is from, say, genetically engineered microbes, and you purify them out, well, you could make phenomenal dairy products, because structurally, these proteins are key to what, gives you, say, a cheese eating experience or a yogurt eating experience. And I was mind blown. I was like, oh, why didn't I think of this? This is kind of like, this is amazing.
What
And it's
year
kind
was
of
this?
intersection, 2018,
Okay.
2018, yeah. So this was intersection of, everything I loved. It was like proteins, and then I started obsessively learning about casein proteins, which are really, really fascinating class of protein. And I was hooked. And so I was like, yes, I'm game. And Matt and I started working kind of remotely while we were in two continents, and really had this idea of having to come together and start a company. And our big dream was to go to IndieBio, which is like a really well-known biotech accelerator here in the Bay Area. And we worked hard to try to get in and have some preliminary data to show that what we're doing is feasible. And then the rest is kind of history, as they say, because like we got accepted to IndieBio, and that really like, propelled us, helped us tremendously, gave us lab and money and place where we could like develop a lot of things in just a few months and raise our first round of funding after. Yeah.
Well, that's a lot to unpack, as they say. Before we move on with other like details of the company itself, I also wonder, like, coming from, let's say, not a whole career in academia, but you did do a postdoc. And when this sort of random guy messages you on LinkedIn, and even though, like, of course, the industry was interesting to you and you had like the technical background he was looking for, um, how did you assess the decision, as an academic person, like, oh, now working at a company, not only working at one, but starting one from the ground up?
Yeah.
Like, was that ever something you thought very deeply about or had doubts about?
Yeah. Yeah. So actually, as I said, I don't think I was ever a true academic at heart or a good academic. And so even in my PhD and then the short postdoc I did after, I actually was always, while I worked really, really hard and I loved what I did in the lab, I spent a lot of time outside of the lab working on other problems. And I actually worked with two, three other startups and NGOs as well, like social enterprises. So projects that are not for profit, various types of projects, actually mainly in developing world. So I really wanted to always be an entrepreneur. I did want to start something of my own. And I was exploring different areas of where I could do that, where that could be helpful. So I definitely had that bug, in me. And that was part of my PhD. I did a lot of side projects, as I said, in my spare time, kind of helping ecosystem in Cambridge that was in social entrepreneurship. So that part was there, and then second part that you asked, like, how did I assess the decision? And well, at the time, actually, I didn't share this part, but as I say, when I left academia, I actually went into consulting and into kind of pure business. I worked for 2 years in London in consulting that's related very heavily to technical work, so data science, data related. But still, I was consulting companies from all around the world on all around topics, all different kinds of topics. And I really learned a lot in that period. It was very dynamic. It was very exciting. And my company was amazing, really supportive, very fun, kind people to work with. But they did know I was very honest. I'm very honest and transparent as a person that like after 2 years, I really got bored. This wasn't for me. I missed science. I missed impact. I missed something like, very different. That I could be doing, where I could be using science and scientific tools to develop something new and better in the world. And so I was looking for a new job at the time and kind of wanted to move into some climate related field for sure. And I didn't know what it was. And so when I was in this job search is when Matt contacted me. So that was kind of like a very again serendipitous moment that it all just like aligned perfectly because this idea didn't come to me despite all of my background that he had this idea because he was very, very passionate about future of food. So yeah, so it was kind of like a great merge of those two things happening. So for me, I have to be very honest. Looking back, it sounds a bit crazy maybe, but I didn't probably very rationally assess this decision because rationally it was kind of crazy. Like I had to call my parents and tell them, hey, I'm actually quitting my job and moving to another continent to try to start this company. And they're like, what? What are you doing? And my friend, everyone thought it was kind of completely insane, to be honest, because it was literally dead. It was like nothing else at the time. It was an idea, mostly with a little bit of data. And so I really just followed my intuition. I followed my heart very strongly. And I learned that that really served me well in life. And when I start overthinking and analyzing and having like rational decision making, it often interferes with what my actual path is.
I can relate to that. But on the data side of things, you mentioned, well, I had not heard of this idea before, but I had obviously learned about different industries as a consultant and so on. Like, through that lens, how did you start to study the economic feasibility of this idea at the time, right? Because we all know like abundantly clear that the easy part is doing this at a bench scale, and then scaling it is a whole other story, especially when it comes to something like food. So, what were some of those first, perhaps, metrics that you were looking at, or maybe models that you were, you know, checking?
Yeah, I must say, I really like your questions. They're very different to any interview or podcast I had, in a very good way, in a very good way.
Sure.
So you're absolutely right. That's something we needed to assess. And while we were at IndieBio, that's when we actually did that work for the first time properly. Of course, that I had to, sense check it before to know that I'm committing my life to something that's feasible, right? That's possible. And the way that I sense checked it in the early days was, okay, are we, doing this with because people have various ideas, right? Some people are trying to use mammalian cells to secrete milk and all kinds
Right.
of things. And like, are we doing this with mammalian cells or with microbes or, or plants? And turned out very quickly that mammalian cells would just not be viable financially. Like that would be so expensive, just maintaining and growing the culture and then never having been scaled something like that actually. Whereas microbial fermentation has been for 50 years routinely scaled and many ingredients have been made that way. So that was the first immediate, proof point of, okay, like if we do this in microbes, this should be feasible. And I looked at molecules such as insulin, or chymosin, the enzyme used in cheese. There are two proteins that are actually peptide and protein that are made same way. They're made recombinantly from genetically modified microbe at large scale at very low cost. So that was enough for me in the early stage to feel like there's definitely precedent. This is definitely possible. And then when we were at IndieBio, we of course started understanding and looking at, well, what will be those drivers of cost, as you mentioned. And really you look at it, as in like three key categories, I would say. One is like, what are the metrics of your process, the product, the efficiency of your process. You can think of it as like a car has an engine and, how efficient is this engine maybe in burning fuel. So same as with microbes producing protein, we have to look at how efficiently they transfer sugar or carbon and nitrogen source into protein. How much can we get for what we put in what time? That's really key. And then how much can we purify out? Because we don't use this big mass at the end. We have to use very nice, purified, clean white protein. So like, what is the efficiency of that purification? And of course, you have to put some assumptions on the numbers, like what are the numbers that we could be hitting? And you can look at literature, you can look at also talking to people. We talk to a lot of people in industry because a lot of these things in industry, they're kind of secret, people don't publish them. So we
Yeah.
have to understand what's possible in industry because it's much different than what's just published in literature. And then second category of cost is all of your materials, all of your raws, right? Like how much, literally just as if you're buying food. So it's like I'm buying materials for fermentation. I'm buying materials for what called downstream processing, so purification. Like how much does that cost? And then the third category is, well, how am I gonna produce this? What is the manufacturing method, facility? That requires instruments, equipment, capex, capital expenditure. But it could also be model where you just work with existing manufacturers and you rent their facility, and then it's more like you pay operational expenses, OpEx. You pay the labor, you pay the work that they do, right? But these are the three key things that we had to assess while we were at IndieBio, and we were very quickly convinced after again speaking to a lot of people and who are in the industry and trying to get these numbers. We were very quickly convinced that yeah, absolutely, we can get to commodity cost, should I say, commodity cost for making our animal-free casein protein. And it was, clear pretty quickly what are some of these key drivers will be. Now, does it make it easy? No, these numbers are difficult to hit, all of these different technical metrics and the way that you can negotiate your contracts with manufacturers, it all comes together from, again, how well you design the process for it to fit facilities easily, all these things. So, but yeah, we absolutely did this assessment in early days, and we knew that, yeah, hey, absolutely something we can achieve, within a reasonable time frame for biotech, which is say 5 to 10 year timeline, and that's where we're getting there. We're definitely slowly on that path.
Yeah, I'm always very curious since you mentioned the path to getting to commodity prices. Um, in terms of the role of the CSO or CTO in that, which is often a role that not a lot of people hear about outside the biotech industry, like CSO. How does that look like for you? What kinds of educational backgrounds you recruit for this?
I must say that when I just started my job, when I started company, I didn't know this. I know I would know it. If I started second company, I would immediately kind of have a footprint. But we had to together, my co-founder and I had to kind of go and just meander and find all this out. And it was kind of slow and painful. And what really, really helped is again the community, because being part of IndieBio, it meant that there were so many startups that went through IndieBio that were older than us, more experienced than us, and already went through a lot of pain. And we could just call them, and they were really kind and helpful, and they would, answer a lot of our questions. And that I do that now. I also pay that forward to other companies. So for all those questions, we would immediately ask someone else, like, hey, so to your point, like I don't come from a bioprocess background. I'm not an engineer. It wasn't obvious to me as someone who's more like a molecular scientist, um, how complex is the fermentation and the downstream processing, how much knowledge and work outside of my domain, that is. And so it became obvious that, well, we really, really ideally do need to have someone who has done this before and who is an engineer, ideally people who are a chemical engineer with, chemi background, and who have before been through process of taking process from bench, bioprocess from bench, through piloting, optimization, and then all the way to manufacturing and delivering to customers, right? So that informed, of course, some of our key hires, like Benita, who joined us, who was for years before at Emirates, a really really great biotech company here in the Bay. Um, she came to lead our bioprocess team and engineering team, and was exactly someone who had that background, everything that I missed, right? So we're kind of complementary. And then on the other hand, someone who would work closely with me and say lead more of all other areas of R&D, like the strain engineering and building tools and methods, assays, analytical chemistry, data. We hired Josh, who came to us from his, again, years of experience at Bolt Threads, where he led their R&D working on animal-free silk proteins. And so it was really, really important to have, to bring in people into the team who have been at startups before, who have had this experience before as well. At least that was my learning and how I found us to be successful. And of course, the entire team that we hired, we were really, really intentional about every single person that we brought on the team, from our like earliest, scientists, earliest people who are still with us today. But it wasn't always perfect. It was, sometimes you make 5 steps forward, 2 steps back, and sometimes it's hit and miss, and then you learn and you correct on the way. And I think the key is also having that mentality of being agile and pivoting and adapting. Like if you are very, very rigid and you just think you know exactly what you're doing, it's great. It's really important to have a plan and to have a vision. We do that routinely, and we challenge that routinely. And it's important to trust yourself, but at the same time, it's important to be open to understanding, you know, when you don't know something. So I would say for me, yeah, the things that really helped, as I said, to summarize, were learning from the community, learning from other people who have done this before, and then understanding what are the technical areas where I have gaps, where I cannot, make right decisions. I don't have good enough knowledge and background, even after reading 5 papers. And so bringing in people into the team to really to have like this kind of diversity and cover everything together.
For those who don't have enough context on your product and your process, because I missed that before, could you tell us a little bit about that? As I understand, you use, as you mentioned, microbial fermentation to produce this animal-free casein, from which you get a powder, the casein powder, and then you mix it with some plant-based fats to make a sort of milk and well, other ingredients as well, from which you make the cheese. But I don't know how many details you can provide about like perhaps yeah, that process.
Yeah, well, you got it perfectly correct. So that's exactly what we do. So you can, represent the company already. But yeah, it is in the nutshell. Of course, then there's million details behind that. But yeah, so we had to figure out like what, class of what kind of strain, what microbe would be best to engineer to produce this protein. And then we also had to figure out what casein target we want to make, because where things get really complex is that, and we have to oversimplify them, that's what engineering is that in nature there's, many hundreds of different casein proteins in various milks of various mammals. And every mammal has, at least four key types of casein, and there are many, many subgenotypes. And so, and you cannot, it can be a cool and exciting research project to try to make all of these, and reconstitute something in vitro. But it's not a good business idea. You want to have something really simple, cheap as possible, right? And so for that, we wanted to figure out, well, can we just, how can we make just one casein protein? That's enough, where with this one protein, you can turn it directly into these products. And that took a lot of work, of course, figuring out, because for us, as I say, we're not just casein company, we're actually a product company. We're very focused on cheese. We're obsessed with cheese, and we want to know how can we make a great tasting cheese that doesn't come from animals. So that was the important part of figuring out. And then the way that the process actually works is, as I say, you optimize your microbe. You want it to produce as much of this casein protein target as you can in a fermentation process that you have to also fully optimize. And you want to make sure that, you feed least cheapest food to it and that it can produce this protein. After the fermentation, you end up with something that looks like a soup, but you don't want the soup. You want just pure casein. And so this is a really complex part as well, because casein falls into a class for, I don't know, who the audience is. Maybe it's very broad, but for
Some
those
of
who
them
are
will be
in
technical,
biology,
so they'll get
yeah,
you.
exactly. It is a class of intrinsically disordered proteins. So, most proteins in nature actually have what we call fold or a structure. They have a very, very predicted and predictable structure, and 100 years of work behind us is just done in developing methods like crystallography, and then later now electron microscopy, very popular to solve these protein structures. And really, a lot that we know about proteins comes from their structure, and then of course their biochemistry, their function. But caseins are not structured proteins that way. They're intrinsically disordered. What that means is they're a little bit like unraveled spaghetti. They do have temporary secondary structures, but they really, really, really depend on their environment, basically. They're they're molecules that like everyone and everything. So, and that makes it hard because they actually have hydrophilic parts, hydrophobic parts. They like to bind to everything. They stick to everything. They're really hard to separate from anything, basically. And so that's the challenges in the recovery and purification process, is how do you purify this protein cheaply, effectively, to really high quality and purity and specification. This goes into food. You have to feed this to people. Like, it cannot smell funky, it cannot have some impurities. It has to have beautiful color. So you want to purified protein while hitting all those specifications, but doing it cheaply and scalably and at scale, obviously, at large scale. And then once you have that protein, we literally mix it. You ask yourself, well, what else is in dairy products? It's some fat, some sugar, some salts. So we actually mix it with everything else that comes from plant-based world, meaning our products are lactose-free because of that, right? We don't have animal sugar. They're cholesterol-free. We don't bring animal fat. And then from there, we kind of treat it like you would for dairy milk, for example. As you said, we can treat it in different ways, process it in different ways to make dairy products. Because if you look at cheese, our main focus, but also yogurt, many other dairy products, they are they are processed products. There's a lot of talk about processed food these days, and we have to really distinguish ultra processed food, things that are just like made from sugar and are, not healthy, from food that come from food processing. Nearly everything comes from food processing, right? And definitely cheese does. Cheese doesn't come from a cow. Cow doesn't spit out a cheese. It gives milk. So in the same way, we through processing, through temperature, through conditions, we can control the coagulation of this protein and how it turns into a cheese product. And then this cheese product has to, in our case, it goes on pizza. We're very focused on pizza, and we'll be selling into pizzerias. So it has to have all these beautiful characteristics. It's a mozzarella we're making. Oh, it has to melt and stretch and brown and bubble and oil off and have particular mouthfeel and smell certain way and taste certain way. And so that's all what we do as well as a product company. We go, from the strain all the way to cheese. Yeah.
When it comes to upstream, a lot is said about how different bioreactor can be when used in pharmaceutical applications versus food or others that are basically new, right, to this industry. Perhaps you could reduce costs by using I don't know, perhaps a different kind of bioreactor. So do you have anything to share that you guys are thinking about, like questions you're asking yourselves, or something like if you had a magic wand, what would you ask for in this stage?
Sure. So actually, if you look at the entire bioprocess, it starts with a bioreactor with a fermenter, but as I say, then it goes through a series of recoveries and purifications. And that's where we find the biggest difference between, pharma and food and other applications is really in the downstream, I would say. In terms of bioreactors, it is, I don't know, there's maybe a lot of misconceptions about the field because the bioreactor itself, the tank, they're actually in a way the cheapest part of the process. You need, one tank. Yes, you need larger and larger tanks. These tanks are most typically different to beer and wine brewing because we run aerobic fermentations and we have to maintain them clean, so sterile, aseptic conditions. But this has been done and figured out, and we actually can use exactly the same or very similar setups as you would in pharma, and they wouldn't be costly or costing more because the cost again doesn't come from the fact that bioreactor is there, it's built, you're using it. The cost comes from the fact of what kind of operations, first of all, how long do you need to run this? How much protein you can make in what time, right? That's what will drive the cost, in what volume. And then how much of it can you recover and what is the cost of those separations? And so, as I said, on the bioreactor, the challenge typically is really because you have aerobic fermentations. You don't want your process to be oxygen limited. And that can be challenged from older tanks, older bioreactors. They just will not have enough mixing and shearing power. And your what we call oxygen transfer rates, OTRs, might be too low for an effective process. And so that is really where people look at, retrofitting or improving and upgrading. And I would say that's where we are also excited about innovation on, how can you improve the oxygen transfer rate without like bringing in an oxygen tank that you have to pump in, because that is costly at scale and you don't really do it. So like new impeller technology, new mixing technology, that's what's exciting. But as I said, on the cost reduction, it's really mostly on the downstream, because when you make a pharmaceutical protein, say an antibody, you will use very expensive, column purification that are, say affinity-based purifications. There's no way you can do something like that for food. You have to rely on like core biochemistry and phase separations in a way of understanding how to separate these molecules. So you really actually, that's where you have differ from pharma. You do purifications in a different way.
Personally, I haven't been following that, specific space like downstream technologies for food applications, but I wonder if there are any new things that you're looking at.
No, well, actually, this is where it really the good thing to do is to not to innovate,
Hmm.
the thing to do is to to use what's existing out there. That is the
Mm.
cheapest way. You just cannot if you the and you know we had to learn this the hard way because yeah, in the beginning we maybe did do things that you know were novel and different, but then were not easy to implement in a manufacturing side. So really what you want to do is you want to think of what is any manufacturer going to have? What is the equipment they're going to have? They're going to have, you know, a lot of filtration units, various types of filtration from ultrafiltration, microfiltration, nanofiltration. They're going to have some centrifugations. They're going to have, you know, maybe some ways where you can do all kinds of like heating and cooling and moving liquids from one place to another. Not even all of them not even have dryers. At the end, you need to dry your product. So really the best thing you can do is, again, is to develop your process understanding very fundamentals of how this molecule behaves, how is it best separated from everything else in that matrix. And can I do this on the existing equipment that, you know, most operators will have? Can I do it in a reasonable time, you know? If it takes me months to purify something, that's not good. It's not good for food safety either, because you could start growing microbes in this, you know. How do you keep it clean? So all these considerations, it's really is like fitting and understanding very traditional manufacturing is the way to develop your process that will bring the cost down, right? And this is what is really you know, possible and different, I would say, in making proteins, whether it's for food, cosmetics, or materials. It's different than, you know, cultivated meat, where a lot of it has just never been done before, never been figured out. Like, how do you, you know, okay, even the bioreactor part, but once you grow, say that you figure out how to grow your cells, mammalian cells in suspension, and, you know, you figure out how to grow them really well for a long time. But then there's also like, you know, how do you harvest them? How do you turn them into food? All these various parts have really, they're very different, again, for mammalian system than for microbial system. And again, for mammalian system, the upstream part is the challenge quite often. That's at least my understanding. I'm definitely not an expert in that field. But the challenge is actually, you know, growing your cells viably in very large tanks in large amounts to those qualities, because they don't behave. They're not as simple as microbes, obviously. So the challenges are different. And as I say, for us, I think really is there's an existing pipeline of how you manufacture proteins for nutraceuticals or food or materials. And it's not easy. The question is, can you do it for your molecule? Can you have a microbe that is, you know, able to grow in tanks that exist with existing capabilities? Do you have process that can fit to existing manufacturers? That's the way to move fastest and cheapest, really.
All of this said, how did you make the decision between manufacturing in-house and working with some sort of contract manufacturing?
Yeah. I think for us, this wasn't even necessarily like a big decision or a conscious
Hmm.
decision to make. It was kind of like the necessity of the real world was that would we prefer to have our own manufacturing site that's fully dedicated to us, custom built, that can run our process perfectly, right? Because in that case, it's kind of like this fitting like a, you know, fitting hand into a glove is like your process needs to fit someone else's facility like glove. But if you build your own, you can build it however you want, and it kind of costs similar money. So you could build whatever you want. It would be ideal for your process. But you know, who's gonna give us that money today or 5 years ago? It's like $100 million you need to build a whole manufacturing site. And even if you'd had that money, is that best use of your money that you've, you know, raised from someone? We don't think it is. So we think you really want to prove your technology in existing facilities. You want to be able to manufacture, you know, metric tons of your material, make your product, and then, you know, maybe there will be a case. And then there is actually many other ways outside of investor world, outside of venture capital world. There's project financing. There's ways to get loans to actually build these and pay them back, you know. So one day, yes, we would love to have our own manufacturing, and it would be the only way to be like super efficient, you know, and bring the cost down to undercut dairy prices. But until that point, we actually can, and we are working successfully with existing manufacturers, so in contract manufacturing, all up to very large scales and up to, bringing the cost down to cost parity. And so that is big difference in investment needed in capex in buying, setting up the facility up front. So it's just a necessity, I think, of how the world works as of
Yeah,
today.
I like how you framed it because in the end, as you've mentioned before, you are going from all the way from the strain to the final product, and what you care about is going out there and sell it, right? And so when you do this, I know that a lot of people will be concerned about the environmental impact that this product can have versus others. But of course, not leaving aside the technical specifications and qualities and the mouthfeel and all of these things. But then there's also a third aspect, which I think many people could be interested in, which is how much healthier or nutritional is this compared to, let's say, animal-derived products? And is it even a decision actually to prioritize nutritional aspects versus other things? So, what have you found in this cheese
Mhm.
space?
Yeah, this is an interesting question when it comes to product, right? Because say now we made a protein, we're turning it into these food products. Well, what is this food product? What is the value it adds to people? What is the value proposition? And you're rightfully pointing that you cannot prioritize everything equally. You'll have to go by certain order of priorities.
Yeah.
But at the same time, it is absolutely paramount. You're putting food into people's mouths, into their tummies, again, into their lives. It is paramount that you make food that is nutritious and healthy and that people will want to eat. We can see that by just consumer choices these days. As much as people can claim that they are care about environment and sustainability, and definitely there's a niche consumer who will put that above everything else, majority of people will not. Majority of us will have prioritized ourselves, will prioritize our lives, our health, and it is just kind of like a it's normal in a way that you kind of look closest around yourself and then further and further and further. And so with that in regard, we absolutely have to put nutrition up high as a priority. Luckily for us, it is how to say, it is a property of what we're doing, in intrinsic property of it, that it will be good and nutritious, because we are making a protein that is usually an animal derived protein that has all the amino acids, including the essential ones. So PDCASA1, it's basically when it digests in your body and your amino acids get absorbed, you get all amino acids that you need. Um, and so immediately, so say you asked about animal products, we can compare this to animal-derived product. We can also compare it to plant-based or vegan product, right, which are on the market. So if we compare it to animal product, I would say that our product is extremely similar nutritionally. So you will get similar amount of protein, you'll get similar amount of fat. We're not trying to change on this. We're not trying to now make like a low-fat product. But what is better in it is that it will also deliver a similar amount of salts and calcium and all that. What is better really compared to animal products is that it will not have lactose. And this is a big deal breaker for 40 million Americans being lactose intolerant. A lot of people in the whole world are lactose intolerant. And so this is a big thing for consumers. Second one, as I say, is that there is no, while your body can produce cholesterol from eating, high amount of fat, of course, there is no direct cholesterol in our product because there is no animal fat. So these are some advantages. And then the biggest one being that how this was made, actually. People talk about natural, unnatural, but it's really great to know how your food was made and to have it traceable. And that's what bioeconomy allows you to do. When you grow animals, you actually first of all, it is not natural to grow animals to take milk of younglings of another species. But also there's just no control over how clean the animals are kept, how well they're treated, whether they've been shoved with a ton of antibiotics, whether they've been given growth hormones. Like all these things will come to you through the milk that they will give you. And so clearly our product is much cleaner and healthier that way, and it is always the same. And then when you compare to plant-based or vegan product, Unfortunately, these cheeses are mostly made from starch and fat, so very high starch amount, quite unhealthy, because there's no plant proteins that are yet able to replicate casein well, and probably ever. And so this is where there's massive nutritional gap. Our product will immediately be massively superior because it will have the protein, the real protein from dairy, and you will get, good amount of protein in per serving. And this is the biggest difference comparing to plant-based products, really. So I would say compared to each of the two sides, there's very different but significant nutritional advantage. We like to say, as a marketing slogan, we do like to say that our cheese is for everyone. This is a cheese that brings everyone to the same table, whether you are lactose intolerant, whether you are health conscious, whether you care about sustainability, whether you're vegan, and you want to eat tasty and nutritious cheese, right? Everyone can eat this cheese, really. And that's, I think, difference to the products that are in the market.
I love that you mentioned the traceability aspect as well, which is something that I don't think is mentioned as much compared to sustainability, for instance. But really, you can have control over all of these aspects that end up, providing different value propositions, basically for your product. So, well, thank you so much for sharing all these stories, these insights with the audience. And finally, is there anything you'd like to share as advice for a future bio founder now that you've been going through this journey yourself?
There's a lot, but it's all, get into detail. I think the most important high level message really is, and I tell it to myself always, is to really keep trusting your intuition, to trust your gut feeling, and to learn to live from that, which is really hard for us, especially scientists. We tend to be very analytical, so we tend to be a lot in our brain and in our thoughts. But intuition is something very different. It comes almost from your energy from within, from the gut feeling. It can be irrational, it can be not rationally explained always. Of course, it might align with some rational hypotheses you might make, but it might not. So I think just building that, and that develops from experience, as you build experience, as you go through life. That's why maybe typically people are wiser when they're older. It's just that you've had enough experience to then know what's right for you, what feels good for you, because there's no universal truth really, on how to build a company or what business model works, or so I think that is the key thing. It is really to find what works for you and how can you be your best self in the world and offer something to the world.
Thank you so much. That is very aligned to how I think.
Great.
Well,
Well, thank you, Sophia. That was a lot of fun and very different questions than what I usually get.
really, what kinds of questions do you usually get?
I guess it's usually more about the business and the product. And yeah, I like that. This is definitely more technically oriented.
Yeah, well, I hope the audience enjoys that too.